to lesson 4 of protein synthesis. Let's get started. So protein synthesis is the process by which cells build proteins. They follow these instructions encoded in DNA, and it involves two main stages: transcription and translation. So how does a gene become expressed? So a gene is a segment of DNA that contains instructions that actually make proteins, and this determines the traits and control functions in the body. So in the cells, specifically your animal and your plant cells, your protein synthesis will actually develop right here in the nucleus. This is where it starts in both the animal and the plant cell. And this is where transcription starts. Transcription is the process where a specific gene in the DNA is copied into a molecule called mRNA or messenger RNA. So again, this happens in the nucleus of the cells because that is where all of your chromosomes, if you remember in our previous units, that's where all of our chromosomes are located, and it starts all in the nucleus. From there, our DNA will be transcribed to RNA. And remember, when we look at that, our nitrogenous bases, C or cytosine goes to guanine, T or thymine goes to adenine, adenine goes to uracil. Remember, RNA does not have thymine. And your guanine goes to cytosine. So C to G, T to A to U, and then G and C. So let's go on to transcription. So with transcription, an enzyme called RNA polymerase, which is that blue circle right in the center of that diagram, attaches to the DNA at the beginning of the gene. So you can see the DNA right here. So it's that gray, light gray strand. So the RNA polymerase will unwind the DNA and reads the sequence of bases that's on the DNA. And those are called the template and the non-template strand. So that dark gray would be your template strand. That light gray would be your non-template strand. So on the template strand, it will read those bases of A, T, C, and G. And it will actually build a complementary pair from the RNA polymerase. And transcription will go from your 5' to 3' direction. So your RNA transcript will be going that other direction. So you can see that your strand, your DNA strand, you can see your 3' is your template strand right here. And you can see that on your RNA, that's right here, it goes from 3' to 5'. So those are those two directions. So mRNA will go from 3' to 5', and your template strand is from 3' this way to 5' this way. So your RNA builds a complementary strand of RNA using your base pairing rules. So remember, A to U, T to A, C, and G. So an example would be if the DNA template strand has the sequence T, A, C, RNA polymerase makes an mRNA strand with the sequence A, U, G. So T to A to U, C to G. When transcription is all completed in the nucleus, that completed mRNA strand will detach from the nucleus in the DNA and leaves and carries the instructions from the DNA to the ribosome, where the next step, translation, will occur. So translation is the process of turning the instructions in the mRNA into an actual protein. This happens in the cytoplasm at a structure called the ribosome. So this is where your ribosome is your protein synthesis. It creates proteins. This is the process that makes those proteins, if we remember in other units. So what is the ribosome? This is just a little review. The ribosome is made of rRNA, or ribosomal RNA, and proteins. The ribosome is like a factory, and this is where all the proteins are assembled for the cell. So your ribosome reads the mRNA sequence 3 bases at a time. So these groups of 3 bases are called codons, where your ribosome will read each codon, and each one of these codes for a specific amino acid. Then tRNA, transfer RNA, is like a delivery truck. Each tRNA carries a specific amino acid. On one end of the tRNA is a group of 3 bases called the anticodon. That's that pink kind of oval-like circle. And these anticodons matches up with the codon on the mRNA. So you can see that first one there on the right, UGG. It matches up with the mRNA strand that is ACC, and it has that protein. So this is your tRNA. And you can remember because it almost looks like a little T carrying kind of like a little oval, and it'll always be carrying these base codes. That is your anticodon, are those base codes. And these little circles up here would be your amino acids. And when they link together with a peptide bond, they form your polypeptide for your protein chain. So the ribosome moves along the mRNA, and it reads each codon and matching it with the tRNA carrying the correct amino acid. The ribosome links the amino acid together in a chain using peptide bonds. This chain will fold into a functional polypeptide chain, also known as a protein. So, a little review. mRNA carries the instructions from DNA to the ribosome. rRNA is part of the ribosome where the instructions are read and proteins are assembled. tRNA delivers the amino acids to the ribosome, matching them to the mRNA codon. And when the ribosome finishes reading the mRNA, the chain of amino acids is complete. So, why is this important? Why do we care about protein synthesis? So this is how cells create all the proteins needed for life. Proteins are responsible for everything from building structures in the body like your muscles and skin to actually speeding up chemical reactions like enzymes. And without transcription and translations, your cells couldn't function and life wouldn't exist. So go ahead and make sure that you finish all of your notes, and thank you for listening.